Hi everyone, my name is Jakub Staroń. This is the next episode of MedChem Essentials, scientific seminars devoted to medicinal chemistry. Today, we will talk about volume of distribution, with the focus on how structural changes affect volume of distribution. Then we will introduce a new concept of area under the curve, and we will conclude with the description how AUC connects with bioavailability. So let's start with the presentation of these two molecules, nifedipine and nimodipine. Both are calcium channel blockers. Both are neutral molecules. Nimodipine is slightly more lipophilic. Volume of distribution is comparable, around 1, and so is the half-life is between 1 and 2 hours. The properties of this drug causes that they need to be administered multiple times a day, almost like approximately every 2-hour interval, which is very inconvenient for a patient. To improve the properties, a basic center was introduced which provided amlodipine. Amlodipine is a basic drug with pKa of 9.5. Volume of distribution jumped up to 17 liters per kilogram, and so is the half-life jumped up to 34 hours. This allowed amlodipine to be administered once a day. Another example can be found within macrolide antibiotics. Erythromycin has one basic center in the molecule. However, assessing by the size of the molecule, the impact of this single basic center on the properties of the drug is quite limited. So the volume of distribution is equal to approximately 1 liter per kilogram. Half-life is 2 hours, which causes erythromycin to be administered multiple times a day. To improve the properties, a second basic center was introduced within the macrolide ring, which provided azithromycin. Azithromycin already has volume of distribution of 33 liters per kilogram, and together with the twofold reduction in clearance, this resulted in the half-life of 69 hours, and azithromycin is administered once per day. Another example of the impact of second basic center of the properties of a molecule can be found within histamine type 3 receptor ligands. Here, those three compounds, compound 1, 2, and 3, all of them have one basic center in the molecule of the same property. And two of those compounds were equipped with different second basic center. Compound 2 was equipped with a strongly basic center, papyridine, and compound 3 with a weakly basic heterocyclic center. So the nature of the second basic center and the impact of those of the nature of the second basic center of the properties of the molecule is clearly visible in the table. So compound 2 has the highest volume of distribution of 94 liters per kilogram and the highest half-life of 250 hours. However, it is not always possible to introduce a second basic center into the molecule. And also, quite often we cannot modify the basic center because that basic center is responsible for the key interactions with our biological target. This is the case of beta-1 blockers. On the slide, we present metoprolol and betaxolol. Metoprolol is a quite polar molecule, moderately basic, with low volume of distribution, 4 liters per kilogram, and quite low half-life of 3.6 hours. In this case, the researchers couldn't modify the basic center because the nature of it was mainly was part of the key pharmacophore responsible for the interactions with the biological target. So what could be modified? Another side of the molecule, the methoxy group, which was extended up to this cyclopropyl moiety. This allowed to increase by 1 log lipophilicity, which caused impact on the clearance and volume of distribution, and also increased half-life up to 17 hours. Now let's move on to the area under the curve. Area under the curve represents the total drug exposure after a single extravascular or intravascular dose. May seem quite complex, but let's describe it. It is in detail. So on the plot, we have changes of the drug concentration after administration with time. First, after administration, the concentration of the drug increases up to the maximum, and then gradually the drug is eliminated from the body, and so the concentration drops. If we divide this plot into time intervals, let's say 1 hour, then a total AUC, total area under the curve, will be the sum of the individual areas. AUC is a key parameter in the preclinical and clinical studies because together with the dose, which is known, of course, we calculate clearance. And once clearance is calculated, we know we have a parameter that represents how quickly the drug is eliminated from the system. So during clinical studies, let's say we have two clinical candidates, two compounds that we want to we introduce into the market, and we don't know which one is better. So for those two drugs, we measure AUC, and then we see, of course, then we normalize it for the dose, and we see which drug has a higher exposure. So the higher compound with the higher exposure usually is the drug that is being selected as the better one, and is being advanced into further stages of the clinical studies. So paraphrasing, AUC is a measure of how much of the drug reaches a person's bloodstream in each period after a dose is given. So you can see when we compare those two drugs, a drug with higher exposure, higher AUC, will be a drug where a higher dose reached a person's bloodstream. This is very important and very useful information to determine the dosing, because from AUC we calculate clearance, and from clearance we know how quickly the drug is eliminated. Because to maintain a therapy, we need to design such a frequency of dosing to maintain a steady concentration of the drug in the bloodstream. And the concentration of the drug needs to be above the minimum efficacious concentration. It needs to be above the concentration required for the therapeutic effect to show up. And also the peaks of the concentration after administration, they cannot exceed toxic levels. So you see, this AUC and the clearance, those are parameters of the essential fundamental meaning for the designing of the efficient therapy using that drug. That parameter is also very useful for identifying potential drug-drug interactions. Some drugs, they can induce or decrease the activity of certain metabolizing enzymes, which can affect the speed of metabolism of our drug. This is very important because in some cases, when the activity of the metabolizing enzymes is being increased, then the concentration of our drug, the exposure, will drop, and we will lose our therapeutic effect. On the other hand, when the activity of the metabolizing enzymes is decreased, then the concentration of our drug and the exposure can exceed the toxic levels. So those interactions are essential to be identified during the clinical studies to design a safe therapy. Now let's move on to the final slide representing the AUC and bioavailability. Bioavailability, it refers to the fraction of drug that is absorbed systematically. We have two types of bioavailability. One is absolute. It compares amount of the drug when administered via non-intravenous form to intravenous form. And for the calculation of the absolute, we use AUC and those AUC measured after single doses administered through non-intravenous or intravenous form. And then we have the relative bioavailability. It compares is used to compare two dosage forms, two usually non-intravenous dosage forms like a tablet or a capsule. So After this comparison, we will know which formulation of the drug provides higher exposure of the drug. I hope everything was clear. So thank you very much for your attention and see you next time.